Hello everyone. Welcome back to our YouTube channel, Equation by Pharmacist Moms. In this video, we will continue our discussion regarding the cell and its functions. Today we will learn 5. Extraction of energy from nutrients by the mitochondria and 6. Locomotion and ciliary movements of cells. Let's begin. Today, first we will learn how mitochondria extract energy from the nutrients. The principal substances from which cells extract energy are foodstuffs that react chemically with oxygen, that are carbohydrates, fats, and proteins. In the human body, essentially all carbohydrates are converted into glucose by the digestive tract and liver before they reach the other cells of the body. Similarly, proteins are converted into amino acids, and fats into fatty acids. Inside the cell, the foodstuffs react chemically with oxygen, under the influence of enzymes that control the reactions and channel the energy released in the proper direction. Briefly, almost all these oxidative reactions occur inside the mitochondria, and the energy that is released is used to form the high-energy compound, ATP. Then, ATP, not the original foodstuffs, is used throughout the cell to energize almost all the subsequent intracellular metabolic reactions. Under the physical and chemical conditions of the body, each of these high-energy bonds contains about 12,000 calories of energy per mole of ATP, which is many times greater than the energy stored in the average chemical bond, thus giving rise to the term high-energy bond. Further, the high-energy phosphate bond is very labile, so that it can be split instantly on demand whenever energy is required to promote other intracellular reactions. When ATP releases its energy, a phosphoric acid radical is split away, and adenosine diphosphate, or ADP, is formed. This released energy is used to energize virtually the entire cell's other functions, such as synthesis of substances and muscular contraction. Next, we are going to study which chemical processes are involved in the formation of ATP and what is the role of mitochondria in it. This diagram demonstrates the formation of adenosine triphosphate, that is ATP, in the cell. It shows that most of the ATP is formed in the mitochondria. On entry into the cells, glucose is subjected to enzymes in the cytoplasm that convert it into pyruvic acid, a process called glycolysis. A small amount of ADP is changed into ATP by the energy released during this conversion, but this amount accounts for less than 5% of the overall energy metabolism of the cell. By far, the major portion of the ATP formed in the cell, about 95%, is formed in the mitochondria. The pyruvic acid derived from carbohydrates, fatty acids from lipids, and amino acids from proteins are eventually converted into the compound, acetyl-CoA, in the matrix of the mitochondrion. This substance, in turn, is further dissoluted, for the purpose of extracting its energy, by another series of enzymes, in the mitochondrion matrix, undergoing dissolution, in a sequence of chemical reactions, called the citric acid cycle, or Krebs cycle. In this citric acid cycle, acetyl-CoA is split into its component parts, hydrogen atoms, and carbon dioxide. The carbon dioxide diffuses out of the mitochondria, and eventually out of the cell. Finally, it is excreted from the body, through the lungs. The hydrogen atoms, conversely, are highly reactive, and they combine instantly with oxygen, that has also diffused into the mitochondria. This releases a tremendous amount of energy, which is used by the mitochondria to convert very large amounts of ADP to ATP. The processes of these reactions are complex, requiring the participation of large numbers of protein enzymes that are integral parts of mitochondrial membranous shells that protrude into the mitochondrial matrix. The initial event is removal of an electron from the hydrogen atom, thus converting it to a hydrogen ion. The terminal event is combination of hydrogen ions with oxygen to form water, plus the release of tremendous amounts of energy to large globular proteins, called ATP synthetase, that protrude like knobs from the membranes of the mitochondrial shells. Finally, the enzyme ATP synthetase uses the energy from the hydrogen ions to cause the conversion of ADP to ATP. The newly formed ATP is transported out of the mitochondria into all parts of the cell cytoplasm and nucleoplasm, where its energy is used to energize multiple cell functions. This overall process for formation of ATP is called the chemiosmotic mechanism of ATP formation. ATP promotes 3 types of cell functions. 1. Membrane transport, as occurs with the sodium-potassium pump, which transports sodium out of the cell and potassium into the cell. 2. Synthesis of chemical compounds throughout the cell. And 3. Mechanical work, as occurs with the contraction of muscle fibers, or with ciliary and amoeboid motion. Upcoming is how cells locomote, and what are the ciliary movements of cells. By far, the most important type of movement that occurs in the body is that of the muscle cells in skeletal, cardiac, and smooth muscle, which constitute almost 50% of the entire body mass. Two other types of movement, amoeboid locomotion and ciliary movement, occur in other cells. Amoeboid movement is movement of an entire cell in relation to its surroundings, such as movement of white blood cells through tissues. It receives its name from the fact that amoeba move in this manner and have provided an excellent tool for studying the phenomenon. Typically, amoeboid locomotion begins with protrusion of a pseudopodium from one end of the cell. The pseudopodium projects far out, away from the cell body, and partially secures itself in a new tissue area. Then the remainder of the cell is pulled toward the pseudopodium. The membrane of this end of the cell continually move forward, and the membrane at the left-hand end of the cell continually follow, along as the cell moves. Now, we will discuss what is the mechanism of amoeboid locomotion. Basically, it results from continual formation of new cell membrane at the leading edge of the pseudopodium, and continual absorption of the membrane in mid and rear portions of the cell. Also, 2 other effects are essential for forward movement of the cell. The first effect is attachment of the pseudopodium to surrounding tissues, so that it becomes fixed in its leading position, while the remainder of the cell body is pulled forward toward the point of attachment. This attachment is affected by receptor proteins that line the insides of exocytotic vesicles. When the vesicles become part of the pseudopoidal membrane, they open so that their insides ever to the outside, and the receptors now protrude to the outside and attach to ligands in the surrounding tissues. At the opposite end of the cell, the receptors pull away from their ligands and form new endocytic vesicles. Then, inside the cell, these vesicles stream toward the pseudopoidal end of the cell, where they are used to form still new membrane for the pseudopodium. The second essential effect for locomotion is to provide the energy required to pull the cell body in the direction of the pseudopodium. Experiments suggest the forthcoming as an explanation. In the cytoplasm of all cells is a moderate to large amount of the protein actin. Much of the actin is in the form of single molecules that do not provide any motive power. However, these polymerize to form a filamentous network, and the network contracts when it binds with an actin-binding protein, such as myosin. The whole process is energized by the high-energy compound, ATP. This is what happens in the pseudopodium of a moving cell, where such a network of actin filaments forms anew inside the enlarging pseudopodium. Contraction also occurs in the ectoplasm of the cell body, where a pre-existing actin network is already present beneath the cell membrane. Next is the types of cells that exhibit amoeboid locomotion. The most common cells to exhibit amoeboid locomotion in the human body are the white blood cells when they move out of the blood into the tissues to form tissue macrophages. Other types of cells can also move by amoeboid locomotion under certain circumstances. For instance, fibroblasts move into a damaged area to help repair the damage, and even the germinal cells of the skin, though ordinarily completely sessile cells, move toward a cut area to repair the opening. Finally, cell locomotion is especially important in development of the embryo and fetus after fertilization of an ovum. For instance, embryonic cells often must migrate long distances from their sites of origin to new areas during development of special structures. Subsequent is control of amoeboid locomotion, that is chemotaxis. The most important initiator of amoeboid locomotion is the process called chemotaxis. This results from the appearance of certain chemical substances in the tissues. Any chemical substance that causes chemotaxis to occur is called a chemotactic substance. Most cells that exhibit amoeboid locomotion move toward the source of a chemotactic substance, that is, from an area of lower concentration toward an area of higher concentration, which is called positive chemotaxis. Some cells move away from the source, which is called negative chemotaxis. But how does chemotaxis control the direction of amoeboid locomotion? Although the answer is not certain, it is known that the side of the cell most exposed to the chemotactic substance develops membrane changes that cause pseudopodal protrusion. In the row is cilia and ciliary movements. A second type of cellular motion, ciliary movement, is a whip-like movement of cilia on the surfaces of cells. This occurs in only two places in the human body: on the surfaces of the respiratory airways, and on the inside surfaces of the uterine tubes, that are, fallopian tubes of the reproductive tract. In the nasal cavity and lower respiratory airways, the whip-like motion of cilia causes a layer of mucus to move at a rate of about 1 centimeter per minute toward the pharynx. In this way, they continually clear these passageways of mucus and particles that have become trapped in the mucus. In the uterine tubes, the cilia cause slow movement of fluid from the ostium of the uterine tube toward the uterus cavity. This movement of fluid transports the ovum from the ovary to the uterus. A cilium has the appearance of a sharp-pointed straight or curved hair that projects 2 to 4 micrometers from the surface of the cell. Many cilia often project from a single cell, for instance, as many as 200 cilia, on the surface of each epithelial cell, inside the respiratory passageways. The cilium is covered by an outcropping of the cell membrane, and it is supported by 11 microtubules, 9 double tubules, located around the periphery of the cilium, and 2 single tubules, down the center. Each cilium is an outgrowth of a structure that lies immediately beneath the cell membrane, called the basal body of the cilium. The flagellum of a sperm is similar to a cilium. In fact, it has much the same type of structure and same type of contractile mechanism. The flagellum, however, is much longer and moves in quasi-sinusoidal waves, instead of whip-like movements. The cilium moves forward, with a sudden, rapid whip-like stroke, 10 to 20 times per second, bending sharply, where it projects from the surface of the cell. Then it moves backward, slowly to its initial position. The rapid forward thrusting, whip-like movement, pushes the fluid, lying adjacent to the cell, in the direction that the cilium moves. The slow, dragging movement in the backward direction has almost no effect on fluid movement. As a result, the fluid is continually propelled in the direction of the fast forward stroke. Because most ciliated cells have large numbers of cilia on their surfaces, and because all the cilia are oriented in the same direction, this is an effective means for moving fluids from one part of the surface to another. Upcoming is, what is the mechanism of ciliary movement? Although not all aspects of ciliary movement are clear, we do know these. First, the 9 double tubules and the 2 single tubules are all linked to one another by a complex of protein cross-linkages. This total complex of tubules and cross-linkages is called the axoneme. Second, even after removal of the membrane and destruction of other elements of the cilium, besides the axoneme, the cilium can still beat under appropriate conditions. Third, there are 2 necessary conditions for continued beating of the axoneme after removal of the other structures of the cilium. 1. The availability of ATP and 2. Appropriate ionic conditions, especially appropriate concentrations of magnesium and calcium. Fourth, during forward motion of the cilium, the double tubules on the front edge of the cilium slide outward toward the tip of the cilium, while those on the back edge remain in place. Fifth, multiple protein arms, composed of the protein dynein, which has ATPase enzymatic activity, project from each double tubule toward an adjacent double tubule. Given this basic information, it has been determined that the release of energy from ATP in contact with the ATPase dynein arms causes the heads of these arms to crawl rapidly along the surface of the adjacent double tubule. If the front tubules crawl outward while the back tubules remain stationary, this will cause bending. Here, we finish our discussion regarding the cell and its functions. We hope that you are finding our videos helpful. We welcome your feedback and suggestions. See you.